This video will talk about the different principles of hemocytometry, which are manual cell counts of white blood cells, red blood cells, and platelets. The general objective of this video is to give an overview on the basic tests or principles of manual cell counting. Now, although most routine cell counting procedures in the hematology laboratory are now automated, it may still be necessary to use manual methods when, for example, an instrument is non-functional and there is no backup for this instrument, or maybe in remote laboratories where instrumentation is not possible, or in a disaster situation when testing is done in the field. Hemocytometry is a process of cell counting, usually done manually. The cells counted are the white blood cells, red blood cells, and platelets. The counting of cells are essential in the diagnosis of diseases and also essential in research. The first and most important equipment used in hemocytometry is the hemocytometer, which is also known as the counting chamber. Manual cell counts are performed using a hemocytometer. The most common one is the Levy chamber with improved Neubauer ruling. It is composed of two raised surfaces, and these surfaces are separated by an H-shaped moat or depression. If we dissect the word hemocytometer, hemo means blood, cyto means cell, and meter means measurement. Therefore, hemocytometer is an instrument used to count blood cells. The hemocytometer is a very thick glass slide. It has two ruled areas at the center, which are coated with metal or rhodium. The H-shaped moat, also known as a gutter or a trough, separates the ruled areas. The raised shoulder or ridge supports the cover glass. Each raised surface of a hemocytometer contains a grid that would be used for counting. These grids are not meant to be visible for the naked eye. Therefore, cell counting using a hemocytometer is done with the aid of a microscope. The hemocytometer grid is made up of different sized squares. The large square, or the primary square, is 1 mm by 1 mm in size. And there are 9 large squares, giving a total of 9 square millimeter. 3 large squares are seen horizontally and vertically, making the square counting area as 3 mm by 3 mm. The 4 large corner squares are used for white blood cell count. Each of these corner WBC squares is subdivided further into 16 secondary squares. The central large square is used for red blood cell and platelet count. The central square is subdivided into 25 secondary squares. And each of these secondary squares is further subdivided into 16 tertiary squares. Only the central large square is subdivided into tertiary squares, giving a total of 400 very small tertiary squares. The central square measures 1 mm. The secondary square measures 0.2 mm or 0.04 millimeter squared, and the tertiary square measures 0.05 millimeters. Just to reiterate, the primary squares measure 1 mm each, secondary squares measures 0.2 millimeters each, and the tertiary squares measures 0.05 millimeters each. This is how the hemocytometer looks like from the side. A cover glass is placed on top of the counting surface. The hemocytometer and the cover glass must meet the specifications of the National Bureau of Standards as indicated by the initials NBS on the chamber. The distance between each counting surface and the cover slip is 0.1 millimeter. This is now considered as the depth used in counting. Thus, the total volume of the entire grid or counting area on one side of the hemocytometer is 0.9 cubic millimeter. There are two categories for diluting pipettes. We have the automatic and the non-automatic diluting pipettes. Automatic pipettes include the Trenner and the UNOPETTE, and non-automatic pipettes are the THOMA pipettes used for white blood cell and red blood cell count. The UNOPETTE is an automatic pipette. It is a capillary pipette that automatically sucks in just the right amount of sample. It is connected to a plastic container containing the exact amount of diluting fluid. These are the THOMA pipettes, and there are two types, the RBC THOMA pipette and the WBC THOMA pipette. Let's now differentiate each from one another. The RBC pipette has a bigger bulb compared to the WBC pipette. The RBC pipette has a distinct red bead, while the WBC pipette has a white bead inside. The total bulb volume of an RBC pipette is 100, while the WBC only has 10. The RBC pipette has a smaller bore compared to the white blood cell pipette with a bigger bore. The upper calibration in the RBC pipette is 101, while the WBC only has 11. As mentioned earlier, dilution will be made to facilitate counting, and THOMA pipettes are graduated diluting pipettes. They have markings on the stem. A 0.5 mark and a 1 mark are seen below the bulb. These are used for the specimen. The markings above the bulb are used for the diluting fluid. If blood is collected at 0.5 and the diluting fluid collected up to the 101 mark, then we can compute for the dilution. 100, which is the amount of fluid in the bulb, divided by the amount of blood collected, which is 0.5, we can now say that the dilution for this is 1:200. The same applies if the blood is collected up to the 1 mark. The dilution for this then is 1:100. For the WBC pipette, if blood is collected up to the 0.5 mark, the dilution will be 1:20 because the amount of fluid in the bulb is 10 divided by blood at 0.5 mark becomes 20. And if blood is collected up to the 1 mark, the dilution is then 1:10. The THOMA pipettes are connected to a rubber tubing used for aspirating blood and the diluting fluid. This was initially meant for mouth aspiration, which is no longer practiced due to the hazards it brings. Instead, a rubber tubing was connected to a syringe to facilitate aspiration. But nowadays, automatic aspirators are connected to the pipettes that gives a more accurate aspiration. Diluting fluids are also used in hemocytometry. Their main function is to disperse the blood cells to facilitate counting. But why do we need to dilute? Remember, there are a lot of blood cells, and counting all of it may be tedious and lead to inaccurate results. That's why we need to dilute it first so that counting can be easier and faster. The first diluting fluid to be talked about is the WBC diluting fluids, and there are three types: 2 to 3% glacial acetic acid, 1% hydrochloric acid, and Turk's solution. One thing that all of these diluting fluids have in common is that all of them are hypotonic solutions made of weak acid. The contents of diluting fluids may be any of the following: glacial acetic acid, 1 normal hydrochloric acid, gentian violet, methyl violet, and distilled water. Glacial acetic acid and 1 normal hydrochloric acid have the same purpose, and this is to lyse mature red blood cells. Gentian violet and methyl violet serves as stains that lightly color leukocytes, permitting easier enumeration. And distilled water is used as a diluent. 2 to 3% glacial acetic acid contains glacial acetic acid, gentian violet, and distilled water. 1% hydrochloric acid contains 1 normal hydrochloric acid and distilled water, while Turk's solution contains glacial acetic acid, methyl violet, and distilled water. The next diluting fluids are used for red blood cell counting. We have Hayem's, Gower's, Toyson's, Bethel's, formal citrate, also known as Dacey's fluid, normal saline, and 3.8% sodium citrate. The advantage of Hayem's solution is that it has no corrosive effect even after an extended period of time. A disadvantage is that it can initiate mold formation and rouleau formation. Gower's prevents the precipitation of proteins in cases of hyperglobulinemia and hemoglobulinemias. Toyson's also initiates mold formation and should be filtered. It has a high specific gravity and contains methyl violet. This is usually used by beginners since red blood cells are easily identified from the white blood cells whose nuclei stain blue. Formal citrate or Dacey's fluid is the best RBC diluting fluid, as it contains a preservative, therefore no mold formation. It also prevents the alteration of cell morphology. Normal saline solution is used in cases of emergency when no other RBC diluting fluids are available. It is also ideal for excessive rouleau formation and autoagglutination of specimen. The diluting fluid used for platelet counting depends on the method employed. Examples of these methods are Riesen-Ecker's method, Guyen-Leek's method, and Brecker and Conkright method. The Riesen-Ecker's diluting fluid preserves red blood cells, which is considered as a disadvantage because the red blood cells may visually interfere with the counting of platelets. In the Guyen-Leek's method, platelets appear as round, oval, or comma-shaped, highly refractile bodies. To facilitate counting, the amount of light in the microscope should be adjusted so that platelets appear as highly refractile bodies. The Brecker and Conkright method uses 1% ammonium oxalate as a diluent which lyses red blood cells, therefore preventing their interference in counting. The platelets appear as round or oval purple bodies and sometimes with dendritic processes. Calculations are needed to adjust to the dilution made and to correct the volume that was used for counting. The general formula for manual cell counts is as follows and can be used to calculate any type of cell count. The calculation yields the number of cells per cubic millimeter. The number of cells counted is multiplied by the dilution factor used. The product of this is divided by the area and multiplied by the depth, which is constant at 0.1. The cells counted refers to the total number of cells that were counted on the hemocytometer. The dilution factor may vary according to the dilution made, but usual dilutions are as follows. White blood cells is 20, red blood cells and platelet at 100. The area refers to the area of squares used for counting. If 1 primary square was used for counting, then 1 square millimeter is used for calculations. If 4 primary squares were used, then 4 square millimeter is used for calculations. The depth is the distance between the surface and the cover slip, and this remains constant at 0.1. An alternative calculation may be as follows. The total count per cubic millimeter is equal to the cells counted multiplied to the VCF and multiplied to the DCF. Now, what is VCF and what is DCF? VCF stands for the volume correction factor. So this is a computation that needs to be done. And the formula is volume desired divided by the volume used. The volume desired is always 1. This is constant since the hemocytometer ruled area for counting gives a total of 1 cubic millimeter. The volume used is a combination of the area and the depth that was used for counting. And this is area multiplied to the number of squares used and multiplied to the depth. Let's say that we are counting white blood cells and 4 large squares were used for counting. So the formula would then be 1, which is constant. The area is the size of the large square used, which is 1. And then the number of squares used would be 4. And we multiply this to the depth, which is also constant at 0.1. The calculations will then be 1 divided by 0.4, making the VCF as 2.5. The DCF is the dilution correction factor, and the formula for this is the volume of the bulb divided by the amount of blood used for counting. The volume of the bulb for white blood cells is 10 divided by, let's say, the amount of blood used is 0.5. The DCF is then 20. The VCF and the DCF are then used in the calculations. Let us now proceed with the principle used in hemocytometry. Basically, the principle is the same for all cells that are counted. It is essentially the same for white blood cells, red blood cells, and platelets. The only difference comes in the dilution made, the diluting fluid used, and the area that is counted. The white blood cell count is also known as the total leukocyte count. The white blood cell count denotes the number of white blood cells in 1 liter of whole blood. This estimates the total number of white blood cells, both agranulocytes and granulocytes. The differentiation of these cells is conducted in another procedure called the differential count. The white blood cell count is a useful measurement in diagnosis. It is utilized to indicate infection and may also be employed to follow the progress of certain diseases and therapies. For the white blood cell count, all cells should be counted in the 4 large or primary corner squares. Start counting with the square in the upper left-hand corner until 16 secondary squares are counted. Cells that touch the top and left lines should be counted. The cells that should be counted are represented by the solid dark circles. And cells that touch the bottom and the right lines, represented by the light circles, should be ignored. We do this to avoid double counting. Let's have an exercise. Let's pretend that this was one primary square you have focused under the microscope. Give the count of white blood cells in this primary square. You have 15 seconds to count. And that is 15 seconds. These are the white blood cells that should have been counted. There are 23 white blood cells in this one corner square. Or there are 23 white blood cells in 16 intermediate squares or secondary squares. Make sure not to count dirt and debris that may be seen on the primary square. After counting 4 large squares, quality control must be made. There should be no more than 10 cell difference for every large square. So let's pretend that these are the number of WBCs counted for every primary square. We have 23, 29, 25, and 20. Are these counts acceptable or not acceptable? Since there is only a maximum of 9 cell difference for these primary squares, this makes this count acceptable. White blood cells on both sides of the hemocytometer should be counted. And as part of quality control, the difference between the cells counted on each side should be computed. The formula for this is V1 minus V2 divided by V1 plus V2 over 2 multiplied to 100. The difference should be less than 10%. V1 refers to the highest count and V2 refers to the lowest count. If the result is more than 10%, then the procedure should be repeated. Again, this quality control checks if there is even distribution among the cells. Let's give an example on how to compute for the acceptable difference. These are the number of cells counted on both sides of the hemocytometer. The first side has a total of 345 cells, and the second side has a total of 338 cells. 345 becomes V1 because V1 is the higher count, and 338 becomes V2 because V2 is the lower count. The numbers are then substituted on the formula, gives us 345 minus 338 divided by 345 plus 338 divided by 2, and all of this is multiplied to 100. This leads to 7 divided by 341.5 times 100. The answer is 2.04%. 2.04% is less than 10%. That makes this count acceptable. Then we proceed to the WBC computation. The formula for the white blood cell count was already mentioned earlier. The general formula is cells counted times dilution factor divided by area times depth. The cells counted should be the average number of white blood cells counted on both sides. If we take the values earlier, that would be 345 plus 338 divided by 2. Dilution factor refers to the reciprocal of the dilution made. If a 1:20 dilution is made in the preparation, then the dilution factor to be used in the computation is 20. The area will be 4 since there are 4 corner squares counted with 1 millimeter square each. The depth is the space between the cover slip and the hemocytometer, which is constant at 0.1. The values are then placed in the formula. We have 341.5 times 20 divided by 4 times 0.1. And we will have 6,830 divided by 0.4. Our white blood cell count is 17,075 per microliter. The alternative for the general formula is the number of cells counted times VCF times DCF. The cells counted is also the average number of white blood cells counted on two sides. DCF is computed with the amount of fluid in the bulb divided by the amount of blood use, so that's 10 divided by 0.5. VCF is computed with the volume desired over volume used, which gives us 2.5, and the depth is constant at 0.1. The values then become 341.5 times 2.5 times 20. Our answer is still 17,075 per microliter. The reference range for the white blood cell count is 3.6 to 10.6 expansion 3 per microliter. The value that we have computed is 17,075 per microliter, or 17,075 per cubic millimeter, or 17 expansion 3 per microliter, or 17 expansion 9 per liter. Our value of 17 is higher than the 10.6 reference range. Therefore, the count that we have computed has an increased white blood cell count. Let's have another example just to make sure that everything is clear. Let's say that these are the number of cells counted on one side of the hemocytometer, and these are the number of cells counted on the other side. The first thing we have to do is to make sure that the difference per square is less than 10. The maximum difference on this side is only 4. That makes the count from this side of the hemocytometer acceptable. And the maximum difference of the cells counted on this side is only 7, which also makes the count from this side acceptable. Next thing we have to do is to compare the total number of cells counted on each side. They should have a less than 10% difference. We do this to make sure that the specimen has been dispersed properly. The total number of cells on the first side is 114, and the total number of cells on the second side is 122. Follow the computation to know the acceptable difference. The difference on these counts is 6.77%, which is less than 10%, making all the counts acceptable. We can now proceed to the WBC count. Get the average of both counts. The average for these counts is 118. Follow the computation for white blood cell count. We will have an answer of 5,900 per microliter or 5.9 expansion 3 per microliter. Following the reference ranges, our count can be interpreted as a normal WBC count. Leukocytosis refers to white cell counts above the reference range, and leukopenia refers to white cell counts below the reference range. Manual red blood cell counts are rarely performed because of the inaccuracy of the count and questionable necessity. The use of other more accurate manual blood cell procedures like hemoglobin and hematocrit are preferred. For the red blood cell count, use the middle large square, which has 25 secondary squares. Count all cells on 4 corner secondary squares and 1 central square. Start on the upper left-hand corner and count all cells that touch the top and left lines. This is how red blood cells would look like under the microscope with a hemocytometer. And this is the central large square where we have to count all the cells. 4 corner intermediate squares and 1 central square will be used for counting red blood cells. This is another picture showing red blood cells under a microscope using a hemocytometer and the squares that should be used for counting. Let's have another exercise. These are the 5 intermediate squares that should be used for counting. Remember the rule of counting all cells touching the top and the left lines. Pause the video and take as much time as you need to count the cells. These are the number of cells counted per square. Square 1 has 97, square 2 and 3 has 99, square 4 and 5 has 102, giving a total of 471 red blood cells in 5 intermediate squares. The same formula is used for the red blood cell count. The cells counted is the average number of red blood cells from 2 sides. These are pretend values of 471 and 475 on each side. If we use a 1:200 dilution, then our dilution factor is 200. Since we have used 5 intermediate squares with 0.04 square millimeters each, our area is 0.2. Depth remains at 0.1. Substituting the values in the formula, it becomes 473 times 200 divided by 0.2 times 0.1. We will have 94,600 divided by 0.02. Our red blood cell count then is 4,730,000 per microliters. A separate reference range is given to males and females. The value that we have computed earlier is 4,730,000 per microliter or per cubic millimeter. It may also be reported as 4.7 expansion 6 per microliter or 4.7 expansion 12 per liter. Our value may be interpreted as a normal red blood cell count. Platelet count refers to the number of platelets in 1 liter or 1 microliter of whole blood. Platelets are difficult to count, even more difficult than white blood cells and red blood cells. They adhere to foreign objects and to each other. They are also small and may be confused easily with dirt or debris. Also, they have a tendency to not be evenly distributed in the mixture of blood and diluting fluid. For platelet count, the central large square is used. All platelets seen inside the central large square or in all 25 intermediate squares should be counted. The same rule applies of counting all cells touching the top and the left lines. In wet preparations like the hemocytometer, platelets are colorless, moderately refractile bodies that are discoid or elliptical in shape. Can you identify what cells are being pointed by the different colored arrows? Which one is the platelet? Remember, platelets are very small. The green arrow points to a leukocyte, while the yellow arrow points to an erythrocyte, and the red arrow refers to the platelet. There are 2 different approaches in counting for platelets. We have the direct and the indirect count. The hemocytometer method is under the direct count. And under the direct count, we can either use phase contrast microscopy or bright field microscopy. Different methods may be seen under each type of microscopy. But among all of these methods, the Brecher-Conkright method is considered as the reference method. In the indirect count, platelets are counted simultaneously with the blood smear, which is less reliable because that means we need to depend on the RBC count. Different methods are also seen in indirect count. For platelet count, all 25 squares in the central square should be used for counting. Same rules apply to platelet count. Both sides of the hemocytometer should be used for counting, and an acceptable difference between these sides should be less than 10%. A platelet count performed from a hemocytometer is verified with a platelet estimate using a peripheral blood smear. The same general formula is used for platelet count. The cells counted are the average number of platelets counted on 2 sides. Values of 350 and 420 are given for this example. If 1:100 dilution is made, then 100 will be the dilution factor. Since only 1 central square was used with 1 square millimeter area, then 1 will be the area used for computation. And the depth still remains at 0.1. To compute for the platelet count, 385 times 100 divided by 1 times 0.1 is 38,500 divided by 0.1. Our platelet count then is 385,000 per microliters. Compute the platelet count using the alternative formula. Calculate for the VCF and the DCF. For this example, 350 platelets were counted on one side and 420 platelets were counted on the other side. What will the DCF be if blood was collected up to the 1 mark? Pause the video and do your computation. VCF is equal to 10, and the depth used is 0.1 in the VCF computation. Our calculation will then be 385 times 10 times 100. Our answer is 385,000 per microliters. The reference range for platelet is 150 to 450 expansion 3 per microliter or expansion 9 per liter. The value that we have taken from the computation was 385,000 per microliter or per cubic millimeter. This may also be 385 expansion 3 per microliter or expansion 9 per liter. Therefore, our value may be considered normal in platelet count. 3 terms are used in relation to platelet count: thrombocytosis, thrombocythemia, and thrombocytopenia. Thrombocytosis is the term used when there is an increased platelet count accompanied by a disease. meaning that there is a coexistent disease that is causing the elevation of the platelet count. Thrombocythemia, on the other hand, is an increased platelet count, but no disease accompanies its increase. Thrombocytopenia is the subnormal number of platelets in the circulating blood. The most common cause of thrombocytopenia is abnormal bleeding. And that ends this video discussing the principles of hemocytometry. Thank you for watching.